Okay, guys, I will come into the first school of Biochimie Structurale, which is structure chemistry. Today, we're not going to go much into depth, but the second time we're going to meet, we're going to go into depth. Yes, today, our first chapter will be based on glucides, which are carbohydrates in short. We're going to look at the introduction, the origin, and répartition et rôle des glucides dans la nature. That is just the répartition is the distribution and role of carbohydrates in nature. Then we're going to look at definition et classification des glucides. Yeah, the definition and classification of this glucide. If you remember the last time we met, I was introducing biochemistry. I gave you a definition of a glucide, or in short, a carbohydrate, of which I said a carbohydrate is a biomolecule consisting of carbon, hydrogen, and oxygen atom. I also earlier stated what a biomolecule is. And we defined a biomolecule as any organic molecule that is present in a living cell. Do you guys remember all this?
Yeah.
Yeah. A carbohydrate is simply a biomolecule consisting of carbon, hydrogen, and oxygen atom, usually with a hydrogen oxygen atom ratio of 2:1. What do I mean? Most of the carbohydrates usually have got a ratio of hydrogen and oxygen ratio of 2:1. 2:1. This is the ratio I'm talking about, 2:1. This is the empirical formula or the general formula of carbohydrates. And carbohydrates are characterized by the presence of a carbon chain carrying an hydroxyl group function. Hydroxyl, this is an hydroxyl group function. Or a carbon function aldehyde or ketone. Carbohydrates are characterized, I will repeat, carbohydrates are characterized by a carbon function, which can either be an aldehyde or a ketone, or by the presence of a carbon chain carrying an hydroxyl group. Aldehyde, this is an aldehyde group. This is an hydroxyl group function. These are some of the characteristics of carbohydrates. Or in other ways, Or in other ways, I can define a carbohydrate to be a biomolecule, micromolecule, which is abundant on Earth. They are commonly known as sugars. I can say a carbohydrate is a sugar. Why are they known as sugar? Because most of them have a sweet taste. Chemically, all carbohydrates are polyhydroxyl. What do I mean? They contain an hydroxyl OH hydroxyl group, which I earlier on showed you here. It's an hydroxyl group. Or aldehyde or ketone. All carbohydrates are hydrate of carbon, and they contain hydrogen. They contain hydrogen, carbon, and oxygen. With the ratio I was talking about, 2:1. We'll see as we go down, we'll see this ratio will be constant in the form of, it will be in the same ratio, 2:1. But there are some carbohydrates which contains nitrogen, phosphorus, and sulfur. Then carbohydrates are denoted as saccharides. And the term saccharide simply means sugars. Are we together?
Yes, we are.
Okay, that's fine. I'll be slow like this. Now, allow me to talk about the origin of carbohydrates. Carbohydrates can be endogenic or exogenic. What do I mean? Endogenic, you get the term endo. Endo means inside, something which is produced inside. Exo is something which is produced outside. Endogenic, they produce metabolically. Exogenic, they produced through the intake of certain food substances. I can give you an example of rice, bread, all those, and so on. Hello, are we together?
Yes.
Yes,
Yeah.
yes.
Sorry? Then we come to the distribution and role of carbohydrates in nature, or importance of these carbohydrates. They play three major roles. Role structurale, raw, metabolic, low reserve. Let's see what these things do. In raw, I'll start with raw structure law. In structure law, these carbohydrates intervene as element to support the cellulose or protection and cognition in the cell. They also intervene as reserve element of plants and animals. In plants, we can talk of starch. You did starch in biology, junior high school, I don't know, it's junior high school, you did starch. And in animals, we can talk of glycogen. You know, the conversion, that mechanism, conversion of glucogen to glycogen. Do you still remember that mechanism? Where energy is being stored as ATP. They also intensify as constituent fundamental molecules such as nucleic acid, coenzyme, enzymes, and vitamin, etc. We come to the raw energetic. In the raw of energetic, they intensify also to say 40 to 50% of the calories provided by human body are carbohydrates. You have to know that 40 to 50% of calories provided by the human body are carbohydrate, which plays a major role in the reserve of energy. The energy is being reserved in the river and muscles in form of what? Glycogen. As I earlier on talked about the conversion, that mechanism we learned in biology, the conversion of glucose to glycogen. When they're in glycogen state, they're being stored in the muscles and the liver. That's what carbohydrates do in terms of raw energetic. And raw energetic and raw metabolic, they simply play the similar law. we go to classification. Carbohydrates are classified in 3 major categories. This classification is based on the number of monomers, monomer unity, and the degree of polymerization. The classification, the first one is classified as monosaccharide, oligosaccharide, and polysaccharide. I think from the prefix itself, we know that mono means one. Poly, it contains plusia. It contains many. Oligo, it's many, but can't be. It is minimal than poly. It's more than one oligosaccharide. So as they are classified, let us see, these are monosaccharide, acidic, these are poly, so to say. These are oligosaccharides, and in this there is also polysaccharide. We'll look at them one by one. Let's look at this monosaccharides. What are monosaccharides? Monosaccharides are simply simple sugars, or this is a basic unity of carbohydrates. They are the fundamental unit of carbohydrates. They cannot be further hydrolyzed. They are non-hydrolyzable, which means they cannot be broken down to a certain degree. As we know what hydrolyzation is simply the breaking down of something, of a particle to a certain degree. They cannot be broken down to a certain degree. Then all carbohydrates, the things you need to know, you need to take note, all carbohydrates consist of all monosaccharides consist of a single polyhydroxyl aldehyde or ketone. Here, an aldehyde or ketone is what we call aldoses or ketoses. Are we together?
Yeah, we are together.
Yeah.
Repeat that point.
Oh. Okay, I'll repeat. Take note of this point. All monosaccharides consist of a single polyhydroxyl aldehyde or ketone. All monosaccharides consist of a single polyhydroxyl aldose or ketose. Are we together?
No, repeat.
And most of the monosaccharides have the empirical formula, which is this one. CHN, don't get confused of this N. It just depends the number of carbon atom. Most of monosaccharides have the empirical formula, this one, CH2O. Then monosaccharides exist as colorless. We can talk of water soluble liquids, etc. And most, I'm talking about the points you need to get note. Most of monosaccharides have a sweet taste. Something you need to know about monosaccharide. Most of the monosaccharides have a sweet taste. And all monosaccharides are reducing sugars. Then, on the basis of the number of carbon atoms here, on the basis of the number of carbon atoms, the monosaccharides may be triose, tetrose, pentose, and so on. 3 means 3, which means that's got a 3 carbon atom. Tetrose means it has got a 4 carbon atom. Pentose is 5. Hexose is 6. Septose 7. Octose 8. And so on and so forth. Are we together? Or maybe I repeat. Hello?
Kindly repeat.
I'll repeat. On the basis of the number of carbon atom, on the basis of the number of carbon atom, the monosaccharides may be named, or they are named according to the number of carbon atom, in other words. And if a monosaccharide has 3 carbon atom, we call it triose. If it has 4.
Excuse me, question.
Yeah.
That formula, does it apply only to monosaccharides or just all carbohydrates in general?
It applies to all carbohydrates in general, but this is mainly based on simple carbohydrates. When you go to complex carbohydrates, you find that complex carbohydrate, they have got certain elements, of which earlier on I mentioned they may have nitrogen, phosphorus, or sulfur. Am I clear?
Yes.
Any questions? Any questions? Then something you need to retain about monosaccharides, though we're going to look at it. We're going to look at it as we go further when we look at the cyclic structure, cyclic structural. But you need to retain this in. I'll remind you. Remember that point. Monosaccharides with 4 or more carbon atom is very complex. Sorry? Monosaccharides with 4 or more carbon atom tend to have a cyclic structure in their nature. This point I'll come and emphasize when you start looking at cyclic structures, maybe in the next school or so. So remember this point. Then I'll give you. Then, oh sorry, I didn't mention this at first. Most of the monosaccharides end with a suffix. We know what a suffix is, huh?
Yeah.
Yeah.
Yeah, most of monosaccharides end with a suffix of O's. When we remove 3 O's, this is a suffix. Most of monosaccharides. Most of the monosaccharides end with the suffix "-os". And the most abundant monosaccharides in the nature is a 6 carbon sugar, which is called glucose. This one, this is the structure of glucose. This is the most abundant monosaccharide in nature. It contains 6 carbon atoms. And the time I was learning this, the time I was learning structure biochemie, our teacher emphasized to know the structure of glucose. And if you take a look at the photo I sent in the group, there are certain things which I highlighted are very much important. You need to know them by heart. Yeah, this is structure of a glucose, which is mostly abundant in nature. Are we together?
Yes, we're together.
Yeah. Then I'll give you certain examples of monosaccharides, which you need to know them by heart. This one, you need to know this: glucose, fructose. These are examples of monosaccharides. These are very important. You need to know these guys. Glucose, fructose, you need to know them. Then ribose, this when you talk about ribose, we're talking about, is it RNA or DNA? Yeah, but yeah, you need to know it as well. But most important, our teacher, the time was teaching us, emphasized about glucose and fructose. If you know these ones, you're well to go. Are we together?
Yes.
Yeah. And simply, guys, biochemie, it's something which requires reading and understanding. So I'm sure as I help you out, explain certain things, you go read out on your own, you'll be able to capture certain things, isn't it?
Yes, yes.
Again, wait. Your vagina is even 4D. To be like, no.
Please, mute your mics.
I don't know.
Hey, tell your friend to mute your mic. Yeah, okay.
My samosa.
You guys, tell your friend to mute her mic. Okay, thank you. Yeah, as I classified the carbohydrates, I said they're classified in 3 forms. We have talked about monosaccharides. Now we're going to the second one, which is oligosaccharides, which is based here, oligosaccharides. Oligosaccharides are sugar polymers which consist of short chains of monosaccharide unit or beer, I can say, which consist short chains of monosaccharides residues. The monosaccharide unity, get me clearly on this, the monosaccharide unity in the polysaccharide chain are linked through a characterized bond called a glycosidic bond. I repeat the point. The monosaccharide unity in the polysaccharide chain are linked through a characterized bond called a glycosidic bond. Earlier on, I stated that oligosaccharides is something which is a mixture of two or more monosaccharides unity. A mono of one. All oligo consists of plusia, not one, but the mixture of something which is more than one. Hence, I'll repeat the point I earlier on stated. The monosaccharide unity in the polysaccharide, poly means many, chain, are linked through a characterized bond called a glycosidic bond. Let me show a glycosidic bond. Do I have a glycosidic bond?
Excuse me.
Yeah, come in.
You are saying oligosaccharides are made up of 2 or more monosaccharides, right?
Yeah.
So does that mean disaccharides are a category that is within oligosaccharides or what?
Sorry, repeat what you said.
You said oligosaccharides, sugar molecules that are made up of 2 or more monosaccharide units, right?
Yeah.
Yes. So my question is is it that disaccharides are a category that is in oligosaccharides, or it's a category on its own?
To answer that question, I'll answer you as I explained down there, of which I'll answer you in the form of most common oligosaccharides in the nature are disaccharide. Are we together? And a disaccharide consists of 2 monosaccharide units. Are we together?
Yeah.
Thank you very much. Are there any questions from the point I raised, which I said the monosaccharide unity in the polysaccharide chain are linked through a catalyzed bond called glycosidic bond. Then I'll talk about, okay, since there are no questions, the number of monomers, the number of monomer unity in the oligosaccharide, sorry. Sorry, wait, classification. Okay. The number of monomer unity in the oligosaccharide usually ranges from 2 to 20. I will show you as I will explain this as I will show you the structures in the following course. Yeah, but as well you can search about this. You see how the structures look. You need to know the structures. Yeah, the monomer unity in the oligosaccharide usually ranges from 2 to 20. Then all oligosaccharide can be hydrolyzed, can be broken down, in short. Are we together? The monosaccharides cannot be broken down because it contains one, leos. Leosidic, these are simply complex. In short, I can say these are simply complex carbohydrates, which may either contains nitrogen, phosphate, or sulfur in them. They only contain carbon, hydrogen, and oxygen. These will contain, they contain other molecules such as sulfur, nitrogen, and phosphorus. Are we together? Then I come back to the point, the question one of your friends asked about the disaccharides.
Yes, yeah.
I'll come back to the question which our brother asked. The most common oligosaccharide in nature are disaccharide. And a disaccharide consists of two monosaccharide units. The most abundant, I'll talk about the most abundant occurring disaccharide is sucrose. Sucrose is simply table sugar. You know what table sugar is, isn't it? Maybe Gulamisas was table sugar, guys.
No, it is.
Yeah, you know
Yes.
it.
Easy about bosses.
I'll say. Sorry, come in. Somebody wanted to say something? Yeah, okay. Let me continue. The most abundant natural occurring disaccharide is sucrose, which is sugar table. Sucrose simply consists of a glucose, is a mixture of a glucose residue and a fructose residue connected. I don't want to don't get lost here.
Just
Connected,
repeat it before we go further.
yeah, a sucrose consists of a glucose residue and a fructose residue connected through alpha 1,2 glycosidic linkage. Don't get worried about alpha 1,2 glycosidic linkage. This is the naming of carbohydrate. When you do nomenclature, you guys, you know how to name these things. Don't get worried at the moment. Okay. Yeah. So what should you retain in your mind is that glucose or sucrose is just a mixture of a glucose residue and the fructose residue. That's what you should take in mind at the moment. Then there's another point you need to retain about oligosaccharides. Oligosaccharides with a 3 or more monosaccharide unity do not occur freely in nature. This point you need to retain. They are usually found or associated with a non-sugar molecule such as a protein. They don't naturally occur in nature, but I'm talking about a oligosaccharide which have got a 3 carbon atom. Usually, they don't actually occur freely in nature. They are found or they are associated with a non-sugar molecule, such as protein and lipid. As glyco conjugated, which we can say we have heard of glycoprotein, glycolipids. I think we have heard all these. We have heard these names, n'est-ce pas?
See.
You have heard of them. Be open if you haven't heard of them. Yeah, okay. We proceed. I can give you an example of oligosaccharides. We have what I earlier on mentioned, which is sucrose, which is a disaccharide of glucose and a fructose, which is a mixture. Di means 2. Saccharide, sugar. So sucrose is simply a mixture of glucose and fructose. Lactose is another example of oligosaccharide, which is simply a disaccharide of galactose and glucose. I can talk of maltose, again, which is a disaccharide of two glucose residue. I repeat my points. Yes, please. Maybe I get it from here. I get it from here. In monosaccharides, we have examples of monosaccharides, which are glucose, fructose, of which I earlier mentioned glucose usually is abundant, one of abundant monosaccharide. Glucose and fructose. These are abundant monosaccharide. Then other examples of monosaccharide, this galactose. and so on. Then I come back to this. I said this one is made up of one, which we said mono means one. Is a simple, in short, is a basic unity of carbohydrates. These are simple carbohydrates, this side. Then when we go this side, we say these are complex carbohydrates, which contains carbon, hydrogen, oxygen, and some other elements such as nitrogen, sulfur, and phosphorus. Are we together?
Yes.
Yeah. Then I went further. On here, I talked about, oh, don't get confused, horocydic and heterocydic. Horo, which means they have a unique chain. I didn't earlier mention. Hectero, this is hecto. This is simply different. They've got a same, different from the prefix. Just get the prefix. Prefix. It will tell us hecto. This is different. Then horo, this is have got a unique. And most of the complex carbohydrates have only a suffix of acidic. Then these have got this suffix. These have got this. These are complex. These are simple. Then dehorocydic. These are two with a unique chain. Polyhorocydic. Poly, this is Prusia, with a unique chain. Sorry, with a different chain. Here I talked about disaccharides, isn't it? Of which disaccharide, I said, I mentioned it in oligosaccharides, which are the most common oligosaccharides. I said the most common oligosaccharides in the nature are what we call disaccharide, and a disaccharide consists of a two monosaccharide unit. And the most abundant, I mentioned, the most abundant naturally occurring disaccharide is a sucrose. And I said a sucrose is a, sorry, I said a sucrose is a disaccharide of, or a mix, simply a mixture of two monomers, which are glucose and fructose, a mixture of these two monosaccharides. That's sucrose. Yes, come in if you want to ask a question.
I have a question. Can you give an example to differentiate the holoside from the heteroside? Because I'm kind of confused.
Okay. For us to differentiate the holoside and heteroside. Hetero simply means different, of which we're going to see it as I go down. We're going to see we're going to see it on here, on polysaccharides. That's when you're going to really get the difference of these two. Okay, okay, or I can answer you in in English, they call it homopolysaccharides. They are composed of only a single type of monosaccharide unit. And I can give an example of such a single type of monosaccharide unit, which can either be starch, glycogen, and cellulose. These are examples of homopolysaccharide. Am I clear? Hello?
I'm still confused.
You're still confused?
Yeah, because I was thinking that since we are talking of homosaccharides and heterosaccharides, so I was thinking that maybe homosaccharide will consist of only one type of monosaccharide. Then the hetero maybe will consist of more than one. Like for example, we have glucose mixing with fructose. So I was thinking that maybe the product will be part of the heterosides. Now I'm confused. So I'm finding it on the side of the other side. That's what is confusing me.
Let me say it in simpler terms. Maybe you get it in this way. When I talk of hetero, these are composed of more than one type of monosaccharide unit. Simpler term, I say they are composed of more than type of monosaccharide unit. They can be a melange of groups, fructose, and so on. Am I clear?
The network was breaking, so we couldn't get what you were saying.
Okay, I'll repeat. Heteropolysaccharide. In simple terms, they are composed of more than one type of monosaccharide. I use different, but put it in simple terms. There's one type of saccharide unit which can either be glucose, fructose, galactose, and so on and so forth. That's a melange, which are different. Then homo, they are composed of only a single type of monosaccharide unit. Am I clear?
Okay, just from there. But when we check, lactose is a mix of two different types of monosaccharides. We are mixing galactose and glucose. So now, how is that connected with what you're saying?
Yeah, galactose, I said it is galactose is a oligosaccharide. Sorry, I've confused. Galactose is a monosaccharide which consists of, remember the empirical formula of monosaccharide.
Yes, yes.
Yeah. I've been earlier stated all monosaccharide have got this formula. There are points I mentioned. And I also mentioned another point. All monosaccharide consist of, remember the suffix.
All right. So what you're trying to say is that for for them to qualify to be homosaccharides, like the consistent or the atoms present in both monosaccharides has to be the same. Like for example, in glucose we have an hydroxyl group. Then when in galactose we have the same hydroxyl group. So when we mix the two, there will be homosaccharides or what?
When you mix the two, which forms di, which there and then change to my oligosaccharide. But for it to be called a monosaccharide, I said it has to have certain characteristic of which I told you earlier on, the time I was starting, I mentioned of certain characteristics of monosaccharide, isn't it? And if you look at, I'll show you the empirical formula or formula built the galactose. You find that it is non-hydrolyzed. It can't be broken down further because it contains one monomer unity. Am I clear?
Yeah.
Guys, are we all together?
Yeah, we are together.
Yeah, then what did I end? I ended on, the badness is that I've seen I'm speaking in English, then you're reading something in French, something can be a little bit confusing you guys. Okay, but yeah, you can also be reading both. Since I wanted to facilitate to make you understand, that's why I decided to do it in English. Okay, these are the basics. If you don't know this, which means you may get lost, isn't it?
Yes, it is. But
Yeah.
the language is just the same.
Sorry?
I'm saying the language you're using is just the same.
Okay. I'll talk about this. I've talked about this. It consists the number of the carbon atom of a monomer. which I said a 3 carbon atom, I mentioned a 4 carbon atom, 5 and 2, 6, I mentioned trios, hexos, a 6 carbon atom, it goes on and so forth. Then I talked about the nature of a carboxylic function, which I said we have got a carbohydrate is identified, is characterized by a carboxylic function, which can either be aldehyde or ketone. Then in French, then in French, aldehyde, this is aldehyde. Ketone in French is cetone. Then in French, take note, a ketone is in English. Cetone is French. Then when you add the suffix cetos, it becomes cetos. You add the suffix of monosaccharide, it becomes cetos. Then on aldehyde, when you add the suffix, this is French, aldehyde. When you add the suffix, it becomes aldose. Are we together? Don't be getting confused when I talk about aldehyde ketone. I'm talking about this scene. I'm talking about this scene, ketone, aldehyde, aldose, cetone. Don't get confused with this scene. Are we clear? Or I repeat myself?
They're clear.
Kindly repeat yourself.
Okay. Earlier on, the time I was trying to introduce what carbohydrates are. I talked about 2 characteristics, of which I said carbohydrates are characterized by a carboxyl function or carboxylic function, which is aldehyde or ketone. Now in French, this is an aldehyde. A ketone in French is cetone. Then when you add, we remove this for you to add the meaning of monomers or the monosaccharides, of which I said monosaccharide. These are simply basic unit of carbohydrates, and they contain carbon, hydrogen, and oxygen. And this carbon and oxygen, they are in the ratio of 2:1. We are going to look at this, don't worry, but I showed you the ratio of 2:1. When you come here, the ratio is, mostly, or I say all monosaccharide have got the ratio of 2:1. We normally have 2 hydrogen and 1 oxygen. That's a ratio of this. Yeah. So for you to add a meaning, it's just like nomenclature. You guys are doing chimie organique. For you to add the meaning to a certain things, like alkanes, you remove certain things. You remove, it's either the prefix or a suffix. Now for this carboxylic function, we normally carbohydrate, we remove this and put the suffix of monosaccharide, and it becomes aldose. Then we add the keton in French it's ceton. We remove this and put the suffix for monosaccharide, and it becomes ketose. Am I clear? Is it clear?
Yeah.
Okay. Then I'll talk about two things here. I talked about aldose and ketose. We're now on the same page. If I say aldose, ketose, we're now on the same page. Okay?
Yeah, oh yeah.
Yeah. When I talk about an aldose, we normally look at the bond, where the bond is formed. For an aldose, the bond is formed on the first. It's formed on the first carbon. For a ceton, it's formed on the second carbon. That's what differentiates aldose from ketose. Are we together?
Can you just explain that again?
Okay. What differentiates an aldose and the cetones? It's the bond. Are we together? It's a bond, where the bond is formed. This one is formed on the first. For aldose, the bond is formed on the first carbon. For a ceton, the bond is formed on the second carbon. Don't get worried. We'll start applying the same rules as we go further, even on naming of these Glucides. There's a rule which you need to apply. Yeah, you need to know for you to name whether it's a beta, alpha. So we'll talk much on these things. Don't at the moment, don't. It's just been introduced to you guys. Are we together?
Yes, we are.
Yeah. So for aldose, remember it's formed on the first carbon. This for ketose, it's formed on the bond is formed on the second carbon. In other ways, do I have a good structure? Yeah. I've got a good examples of simple sugars. Have you seen this for aldose? It's formed on the first carbon. Have you seen for ketose? Second carbon. Hello? Hello?
Yes, we can see that.
Yes, it's visible.
Yeah. So you should take, it's visible. You should take note of this. I'm trying to emphasize, if since my network here is saying it's not stable, I need to know if you guys are getting me. Yeah. This is what differentiates an aldose from a acetone or ketose, or an aldehyde from a ketone in English, so to say. These I've already talked about them. Then we're going to talk about the configuration of the D and L. Remember, I haven't touched about polysaccharide. Yeah, but I'm going to talk about the configuration for you to. As we go further, you get what polysaccharides are in detail. We have to first understand these things. We're going to talk about the configuration of the D and L. What normally determines the configuration of the D and L in carbohydrates is the hydroxyl function group. That's what normally determines the what you see, D aldose. That's what determines this. Hydroxyl function group, I will take you back. Sorry. Hydroxyl function group is these are hydroxyl function group. But not every hydroxyl function group determines the D and L. Get me clear on this. Not all hydroxyl function group determines the D and L. No. There is what we call carbon asymmetry. Do you know, guys, do you know what a carbon asymmetry is?
No.
No.
A carbon asymmetry is simply a carbon which is bonded by 4 different radicals or elements, so to say. What do I mean by 4 different elements? If you see here, it's a carbon. This is a carbon. It has been bonded by 4 different constituents. There is this, and an hydroxyl function. And they have to be, always have to be an hydroxyl function amongst the 4 different constituents. Are we together? For you to determine the D and L, this is the configuration of the D and L in carbohydrate. For you to determine the D and L, I'll emphasize on this, you need to look at the carbon asymmetry. A carbon asymmetry is simply a carbon which carries 4 different constituents or elements or radicals. They say radicals in French. These are the 4 different what? Then if an hydroxyl function is at your right hand, like the way you're seeing it, if it is at your right hand, we say D. If an hydroxyl function is at your left hand, we say L. Are we together? here I repeat myself.
C'est clair.
It's clear?
Oui.
Yeah. Then I'll come back to I want to explain something here. I talked about the ratio. I said most of monosaccharides have got a ratio of what? 2 to 1. In the first place, observe this formula. It has got, that is an hexose. It contains 6 carbon atom. Then we come here. You see this one? It doesn't have any carbon atom. And I said and I said carbohydrates are in the ratio of 2 to 1. 2 to 1, hydrogen and oxygen. In terms of hydrogen and oxygen, 2 hydrogen, 1 oxygen. Then we come back here, get back here. You see this formula? This is a formula for what?
Glucose.
Yeah, exactly. This is the formula for glucose. And if we ratio it down, it's going to give us the ratio of what? Oxygen 2 to 1. You do your division. I'm sure you did a little bit of mathematics. Listen to this. It's 2. It's going to drop down to this 2 to 1. Or you simplify it in short. Am I clear? Yeah. And then I had mentioned of ribose. Yeah. Now it's clear. It's RNA, not DNA. Earlier on, I had said DNA. I was doubting whether it's DNA or RNA. This is ribose is an example of monosaccharide I had mentioned earlier on, if you remember. Yeah. Then I wanted to talk about this is the simplest way of writing our carbohydrates. This is galactose. This is the simplest way of writing our carbohydrates. I wanted to talk about epimer and the likes, but you need to have a background of stereochemie. So I have to give you guys a little bit background of stereochemie. Then that's when you'll be able to understand what an epimer is. Are we together? I can't say in this. I think we'll arrange, then I'll do stereochemie, which was supposed to be done, which are supposed to do it in chimie organique, where we talk about my isomers. You did isomers in secondary school, n'est-ce pas?
Yeah, just a bit of it.
Yeah, that's where we're going to drop into Fisher. Maybe if you want, can we do it, or are you tired? Yeah, you can even see here. This is D. Here I can give a clear example of how to name D and L. Actually, in French, when you see a C and a Y, this C and a Y, I see in here. I see in it here. C and a Y, it means carbon asymmetric. Then here, what determines this, I said earlier on, you need to look at a carbon asymmetric. You come here. Is its carbon carrying 4 different constituents? You verify. It's different, it's different, it's different, it's not different. It's the same with this one. You see? You come here. Let's verify. It's different, it's different, it's different, it's not different. They're the same with this one. You go down. We are searching for a carbon asymmetry on this compound. It's different, yes. It's different, yes. It's different, it's different. Then this satisfies to be a carbon asymmetry. After that, you look at where the hydroxyl functional group is. Is it at the left or is it at the right? Am I clear? Then you say, if it is at the right, you say it's D. Looking at this. Sorry?
How do you tell that the carbons are different? I heard you were saying it's different, it's not different. I didn't know what you were doing.
Oh, do you know that? I hope this is a basic thing. Everyone has to know this. A carbon forms how many bonds?
4 bonds.
4 bonds. So amongst the 4 bonds it forms, for it to qualify to be a carbon asymmetric, all the 4 bonds has to be different. That's why I can simplify carbon asymmetric. All the 4 bonds have to be different. Here you see this bond. Let's take this carbon here. You're seeing where my what is. Let's take this carbon for instance. It's bonded to, we said the carbon has to be bonded to 4 bonds. It's bonded to the first carbon, second hydrogen, third it's bonded to carbon, of which we have reached here. It doesn't satisfy the rule of what? Carbon asymmetry, which is supposed to be bonded to 4 different constituents. Are we together? That's how I was saying it's different here, it's different here. Look at this. Here it's bonded to carbon, and here it's bonded to what? An hydroxyl functional group. These 2 are different. So I said it's different, but here and here it's the same. Am I clear?
Ndipo vapanga sensa pamanje bosses.
Vapanga sensa pamanje?
Then starts over first.
Okay. Chawamanga vapanga sensa. Then we come to acetone. This is aldose. What did I say? Somebody to repeat how you identify this as an aldose. This is acetone.
Yeah, for an aldose, there's somebody else wants to say.
Okay. What you can be raising up your hand if you want. Okay, just come in. One person to come in. Frankenstein. Frank, are we there?
Okay. We see the position of the carboxyl group. So when we have it linked to the first carbon, it is an aldehyde. Then when you have it linked to when it appears in the middle of the structure, then it becomes a acetone.
Okay, thank you. Taonga. Taonga, is Taonga there?
Yes, I am.
Okay. Yeah. How do you identify this is an aldose or this is an aldehyde? This is a ketone or acetone in French.
For an outdoors, you say that there's a bond on the first carbon in the molecule. Then for acetone, there is a bond on the second carbon. Yes, the second carbon.
Exactly. Exact. As we have seen here, it has been denoted. As she has said, for you to differentiate these two, the bond is formed on the first carbon for an aldehyde. Then for acetone, a bond is formed on the second carbon. Am I clear?
Yes, you are.
Okay, thank you. Then we proceed. This one, I'll come and explain about when you do, I'll briefly explain to you guys about stereochemistry, whereby we are going to look at a Kira molecule or chirality. If you want, you guys can research about it. Yeah, this is the carbon asymmetric. You see here it's different to this. C is different to this. These are different, which means it satisfies the condition to be a carbon asymmetric. Then here it's a mirror superposition. We'll look at this when I talk about stereochemie, of which you're supposed to look at, you are supposed to learn in chemie organic, but I'll just open your mind a little bit before you reach there. Am I clear?
Yes.
Yeah. Then here you can take this is a good look of why you can differentiate these two. And one thing you need to take note, always bear this in mind, put it on your heart. A glucose is an aldehyde or aldose. It falls under aldehyde. A fructose, it's a acetone. It falls under a ketone. Are we together? I've emphasized about these two. I will emphasize about glucose and fructose so much. Need to know them. Then you need to know that, and this is the, let me not say the only acetone so far. Yeah, but it's the one which they mostly use in exam question, a fructose. Yeah, you need to know it. Then a glucidic bond on oligosaccharide. I talked about a glue glucosidic bond or linkage. I talked about it, but don't get worried. I wanted just you to see how the glucosidic bond I was talking about. I'll explain. This will come under cyclic structure. When you start doing structure cyclic de sucre, that's when I'll explain this. This is a fructose, of which I said a fructose is a mixture of two disaccharides, which are fructose and glucose. Yeah. From so far what we have looked at, are there any questions?
I've got a question.
Yeah.
Yeah, can you kindly go and repeat on how to identify the D and L, and also how to differentiate the carbon?
Okay. Configuration of the D and L. The configuration of the D and L, how it comes about. Wait, just wait. How it comes about, it's been determined by an hydroxyl function group. And it's also based on an hydroxyl function group has to be based on a carbon asymmetry. And which I said a carbon asymmetry has to satisfy, for us to call it a carbon asymmetry, it has to satisfy one principle, which is it has to be bonded to 4 different constituents. Am I clear? I'll repeat in other languages. In another language, sorry. A carbon asymmetry, as we all know, a carbon has to be bonded to 4 different atoms, n'est-ce pas, or elements. And a carbon asymmetry, for it to satisfy this is a carbon asymmetry, it has to be bonded to 4 different constituents, which are this is different to all these, n'est-ce pas? And another, for you to identify D and L, you have to look for an hydroxyl function where it's bonded to a carbon asymmetry. Am I clear? Always search for an hydroxyl function group which is bonded to a carbon asymmetry. And when the carbon asymmetry or hydroxyl function group is at your right hand side, we put the prefix D. D-glyceroaldehyde. When the hydroxyl function group is at your left, we put L. L-glyceroaldehyde. Am I together? Or are we together, sorry?
Yes.
Is it clear?
Yes, it's clear.
Any questions? From all what we're looking at, any questions? There are no questions. Okay, let me ask questions. Cond one.
Yes, boss.
Yeah. Kindly explain to me what you had researched about glucide or carbohydrate, or what you have understood so far.
All right.
Just a
So,
brief.
what I've understood is that glucides are polyhydroxy products that possess a functional group of an alcohol oxide, then which are the aldehydes and the cetones. The general formula is CN open brackets H2O close brackets N. Then they're classified into monosaccharides, oligosaccharides, polysaccharides. Yeah, and then another thing that I noted was yeah, their roles, their importance, like in terms of the glucides, their importance. Talked about the structural role, the energetic role. So in the structural role,
Just end there. Thank you. Thank you. Fumbani Sichali. Yeah, Fumbani Sichali. You guys, let's interact. Bless. Are you there? Okay.
Right. Okay.
No. Let me just say something. Let me just say something. Just be free to say what you have learned. Just be free to say what you have learned. As you are speaking, you also learn and it to stick into your mind. For these things, have to stick into your mind. You have to keep them at heart. You're going to use them at school. That's what I'll emphasize. Just be. We are here to teach each other, you know. So let's be free. Blaze, you can come in.
All right. I've learned of the three, or the two types of, or the two classes of carbohydrates, which are monosaccharides and oligosaccharides. And under monosaccharides, we define them to be the simplest sugars, which cannot be further hydrolyzed.
Yeah, breaking it down.
Yeah. Then, so we are also given examples under the same monosaccharide, which were they start from 3 going upward. So when we have three carbons in a monosaccharide, we put the suffix
It depends on the number of the carbon.
Yeah.
It depends on the number of carbon atom.
Yeah, like that. Then on the X which has 6 atoms of carbon, we are given examples of glucose, fructose, and galactose. Then when we go to the oligoside, oligosaccharides, we have two
We looked at
groups, which are oligosides and heterosides, something like that. Yeah.
yeah, thank you very much. Just there. Yeah, just continue reading them in French. You get there, you'll be pronouncing the name correctly. Don't give up when you fail to pronounce them. Yeah, that's what I can encourage you. You're heading there. And make sure you revise what we do, what we did today. I'll ask one person. Shingano blessing.
I just give the summary of what I've understood.
Yeah.
Okay, so basically, glucides are biomolecules consisting of carbon, hydrogen, and oxygen atoms. They are of three types. We have monosaccharides, disaccharides, and polysaccharides. Monosaccharides are basically just simple sugars which cannot be further hydrolyzed. So these monosaccharides, they consist of the single polyhydroxyl, aldose, or ketose bond. They have the characteristic of being colorless. They are all reducing sugars and have cyclic structures. With disaccharides, they are formed of two monosaccharides. For example, we have sucrose, lactose, and maltose. And then when it comes to polysaccharides, they are just generally made up of many monosaccharides. We have, I can give an example of this, of cellulose. Yes. Okay. And when it comes to the identification of an aldose and a ketose, we find that an aldose is an aldehyde is identified when we notice that the double bond is formed on the first carbon atom. Then we know it is an aldehyde. When we want to know when we want to identify a ketose, we will notice that the double bond is formed on the second carbon atom. Yes. That's
okay. So. Thank you very much for the summary.
You are.
We've got Lillian. Lillian, what did you understand?
Okay, what I understood is glucides.
Don't talk about glucides. Talk about the configuration. Try to talk about the D and L configuration.
Okay, what I understood is for you to determine whether it's a D or an L configuration, you look at the carbon asymmetry, and this carbon asymmetry is a carbon atom bonded to at least 4 different, not at least, okay, it's bonded to 4 different. It must be, yeah, bonded to 4 different constituents.
Yeah.
Yes. So when we find that the hydroxyl group is bonded to, is it the right? Then that's a D configuration thingy. And then when we find that the hydroxyl group is bonded to the left, that's an L type of configuration. That's what I understood.
Okay. Thank you very much. I've seen we're getting somewhere. Yeah, you guys understanding. And feel free to interject me whenever you don't understand. And let's make it a live discussion, so to say. Then I wanted to talk about isomery, the which are isomers of monosaccharide, I'll say it in English. Isomers of monosaccharides. I wanted to talk about isomer of function, my epimer, diastrome, diastroisomer, enantiomer. I wanted to talk about those things today, but I think you guys, we can arrange a time. You tell me the time you'll be free. We look at these things which are under stereochemie, which you're supposed to do in organic chemistry, such that when you start doing them in organic chemistry, it will be a revision to you guys. Then that's why we'll look at Fisher as well at the end of the same thing. So you guys, you just tell me, because the next school I plan to do structure, cyclic structures. We look at the properties, physical and chemical properties, and certain reactions. Yeah, so you guys, you tell me. Actually, I will use something in English to further. Or that mirror you are seeing, well, I'm going to explain it here, you guys. Yeah, you can also take a look. You can also research about stereochemistry. That's where I will start from. I think I'll just make it a core of this to broaden up your understanding, you guys. Otherwise, thank you for your participation. Yeah. It was so nice to be with you, or to discuss with you guys. If you have got anything to say, you can come in before I leave. And if you haven't understood anything, you're free to DM me to ask a question, I'll respond.
Thank you for the class.
Okay, and you guys, you must start studying it. This thing will be getting bulky. Kindly start studying. As you study, have fun, play, and the likes. Yeah. We'll meet next time, you guys. And if you have anything to say, I'll emphasize any query, you can inbox me. You have my number. Yeah, otherwise, continue having a productive day.
Get us on both.
Yeah. Kundwan, you wanted to say?
I was saying, get us on. You had said you are not feeling too well in the beginning.
Thank you. Yeah. I'm not feeling so well, so to say. I'll be fine. All
right.
You just know the heat ya kuno ya kwatu.